permeability. So what will we be learning today? Today's lesson will cover the structure of the cell membrane, how it is involved in selective permeability, and how it separates the internal environment of the cell from the external environment, the fluid mosaic model, and what type of molecules can pass freely across the membrane, and what type of substances need some help with embedded channel and transport proteins. And finally, we'll look at cell walls and how they form boundaries. So let's begin. So as you recall, the structure of the cell membrane is made up of phospholipids, and these phospholipids are amphipathic. Well, what does that mean? Well, that means they're hydrophobic and hydrophilic at the same time. These phospholipids spontaneously form a bilayer in an aqueous solution, as shown on your screen. The fluid mosaic model is this moving phospholipid bilayer composed of molecules such as proteins, steroids, and carbohydrates. The selective permeability of the cell membrane is the direct consequence of this membrane structure. So what things get across the cell membrane? Well, small nonpolar molecules are able to pass freely across. What are examples of those? Nitrogen, oxygen, and carbon dioxide. So what happens if they can't get across the membrane? Well, hydrophilic substances such as large polar molecules and ions cannot freely move across the membrane. They need some help, and that help comes through transport proteins. There are two types of transport proteins. First one, a channel protein, which is a hydrophilic tunnel spanning the membrane that allows specific target molecules to pass through. The second one is a carrier protein, which spans the membrane as well and changes shape to move a target molecule from one side of the membrane to the other. Small polar molecules, like water, can pass directly through the membrane in minimal amounts, but it's really, really small. So what is the purpose of a cell wall in some cells? Well, it's a structural boundary and permeable barrier. As a structural boundary, as in the case of a plant cell, it protects and maintains the shape of the cell. It prevents against cellular rupture when internal water pressure is high. It helps the plant stand up against the force of gravity. Now, as a permeable barrier, things can't get through, but the way they get through is through these small holes called plasmodesmata, which are between plant cells that allows for the transfer of nutrients, waste, and ions. However, one thing I do want to point out is do remember that animal cells do not have these cell walls. The cell walls are composed of complex carbohydrates. You will find cell walls in plants, and they're made up of cellulose, which is a polysaccharide. You'll find them in fungi, and they're made up of chitin, which is another polysaccharide. And finally, you'll find them in prokaryotes in the form of peptidoglycan, which is actually a polymer consisting of sugar and amino acids. So let's practice what we learned today. Today's skill focus is on statistical test and data analysis. Use data to evaluate a hypothesis or prediction, including supporting or refuting the alternative hypothesis. So let's begin. What I would like you to do is you're going to pause your video, you will analyze the table, and you will read your question. Once you feel comfortable, select your answer and then push play again. Welcome back. For this question, you were looking at which of the following identifies the treatment group most likely to have provided an unreliable estimate of the mean and correctly explains why the estimate appears unreliable. So your answer should have been A, treatment group 2. The reason for that is the lower than expected mean absorption compared to the rest of the treatment groups and the higher standard error found in the table. So what should you be taking away from this lesson today? The structure of the cell membrane results in selective permeability. The selective permeability is a direct consequence of membrane structure. Small polar molecules, including nitrogen, oxygen, carbon dioxide, can freely pass across the membrane. Hydrophilic substances, however, such as large polar molecules and ions, need to move through embedded channel and transport proteins. polar uncharged molecules, including water, can pass through the membrane, but in small amounts. The cell walls provide a structural boundary as well as a permeability barrier for some substances to the internal environments of the cell. The cell walls of plants and prokaryotes and fungi are composed of complex carbohydrates. Well, I hope this video and lesson was informative to you. Until next time, thank you.